Hello and welcome. Today we will be introducing you to synthetic modified mRNA treatment for adult cardiomyocytes. Let's meet the team. Hi, I'm Curtis. We have Naomi, Carmen, Gonzalo, and Eddie.
Let's start. For a long time, the existing consensus involved the concept that the heart is incapable of regeneration. Once the heart has developed, cardiomyocytes lose the ability to regenerate in case of lost cells or damage, such as myocardial infarction. Approximately 1.5 million cases of myocardial infarction occur annually in the United States and is the leading cause of death in the world. It has been shown that cardiomyocytes can re-experience the process of cell division through mitosis after suffering a heart attack. Several strategies have been proposed for the regenerative cardiovascular therapy. However, there has been no significant feasible target clinical strategy for treating heart failure until recently. Manipulating cardiac gene expression in a rapid dosable manner and make it tissue-specific without genomic integration would represent a major advancement in heart failure therapy. Recent studies have shown that using modified mRNA encoding specific factors, such vascular endothelial growth factor, result in the expansion and direct differentiation of heart progenitor in murine myocardial infarction models. Results from this study suggest that modified mRNA is a versatile approach to enhance long-term organ repair.
The heart has a limited capacity to regenerate the loss of cardiomyocyte due to an event such as a myocardial infarction. Synthetic modified mRNA is a new gene delivery vector that is increasingly utilized in the vivo application. Modified RNA is a relatively stable, non-immunogenic, highly expressed specific molecule that has been shown to mediate high and transient expression of proteins in different types of cells and tissues. Synthetic modified mRNA therapy can induce the activation of adult cardiomyocyte proliferation, attenuating the innate and adaptive immune response and inducing angiogenesis. It can also prevent cardiac cell apoptosis and necrosis. So far, modified mRNA therapy is a promising curative method for cardiomyocyte regeneration. mRNA routes of delivery. There are several ways to introduce exogenous proteins to the cells of a living organism. The most straightforward way is to directly inject the desired peptide or protein to the body. The first studies using RNA as a method of gene delivery and protein overexpression involve the injection of naked RNA. This approach maintains high levels of control over protein dosage with immediate effects and with no need for cellular translation. An effective cardiac gene therapy delivery platform must have the following attributes: ability to transfer the gene to different areas of myocardium including infarct, border, and remote zones. Transduce sufficient numbers of myocytes of left and right ventricles. And establish a predictable relationship between the amount of cardiac genome copy, number, and cardiac function. During direct myocardial delivery, mRNA is exposed to various critical extracellular and intracellular components. For delivery systems, mRNA must navigate through the extracellular matrix in order to reach cardiomyocyte's plasma membrane. This process is regulated by tissue collagen and hyaluronic acid. After passing through the extracellular matrix, mRNA must be taken up by cells via receptors or non-specific binding mechanisms. Nucleic acids typically enter cells through endocytosis, phagocytosis, scavenger receptors, or micropinocytosis. The cellular endosome is one of the most difficult biological obstacles to overcome.
To improve the stability of mRNA in physiological conditions, cell penetration, and endosomal escape, mRNA can be complex with biomaterials. Lipid nanoparticles are very efficient and widely used for in vitro transfection.
We read some articles about the clinical approach of using purified vascular endothelial growth factor A mRNA as a therapy that resulted in the improvement of cardiac function after intracardiac injection 1 week post-myocardial infarction. The researchers' team screened for transfection of protein expression efficiency using purified vascular endothelial growth factor alpha mRNA in a secret selling buffer injected into the heart of the animal model. After just 1 hour, there was heterogeneous cell uptake of mRNA and protein production. They identified the mRNA marker on the cardiomyocyte cell membrane. The researchers then did an intracardial injection across the infarct area after the induction of a myocardial infarction in the animal models. Surprisingly, 2 months after the injection, they observed significant improvement in several cardiac systolic parameters. The recopilated data demonstrated a prolonged expression of up to 192 hours, which is able to limit cardiac infarct expansion and fibrosis that resulted in the improvement of the systolic function after 1 week of a myocardial infarction.
Recent research showed evidence in animal models of the therapeutic use of modified RNA as a possible treatment of myocardial infarction. The intramyocardial injection of synthetic modified RNA encoding human vascular endothelial growth factor is a very efficient angiogenic. One of the roles of this factor is to act as switch of cell fate in human cardiac progenitors, resulting in the expansion and direct differentiation of endogenous heart progenitor in a murine myocardial infarction model. The vascular endothelial growth factor A modified RNA resulted in the improvement of the heart function to enhance the long-term survival of recipient. One of the functions of vascular endothelial growth factor A is the stimulation of new blood vessels and in this way help replace the damaged cardiac muscle. It is important to emphasize that this protein is naturally secreted by different types of cells. Previous studies in animals of vascular endothelial growth factor A have obtained negative results due to antiviral immune response after induction of this factor using naked DNA plasmid, recombinant protein, and modified virus. The control of vascular endothelial growth factor A expression is critical for good therapeutic efficacy. Studies in independent groups have shown that a prolonged exposure of vascular endothelial growth factor A causes an excessive vascular permeability. Vascular endothelial growth factor A modified RNA has in several studies shown a greatly diminished immunogenicity when administered directly to the skeletal muscle. The DNA plasmid must be administered controlling the rise of interferon beta and RIG1. Among other modifications to avoid an immune response are to optimize selection of nucleotide transcription, UTR sequence, and purification. This modification, in addition, limit the expansion of myocardial damage and fibrosis. These results go hand in hand with other studies that show low toxicity to vascular endothelial growth factor A modified RNA. These results together show that this therapy is an efficient and practically not toxic approach for the delivery of high-efficient genes in the cardiac tissue.
As we know by now, the typical consensus for patients that have suffered an MI is that the heart is incapable of regeneration. However, recent studies have shown that with the use of modified mRNA therapy, adult cardiomyocytes can be reintroduced into a regenerative state. Although this is a very promising therapy and some have been approved for human trials, mRNA therapy for cardiomyocyte regeneration still has several challenges to address. Possibly the biggest challenge for this type of therapy is trying to figure out a proper dosing method or somehow introducing a limiting factor to prevent the mRNA from remaining in the tissue indefinitely that may lead to prolonged expression of the genes and thus undesirable long-term effects such as uncontrolled proliferation. Thanks for listening. Hope you enjoyed our presentation today.